Scientists write papers to communicate their findings. Other scientists then read and discuss these papers. This is a recording of two biophysicists candidly discussing a research paper.
Hello and welcome to episode 31 of the Lemma Journal Club.
I'm Linnea, and with me, as always, is Bezia.
We have a terrific paper for you today entitled Ribosome Molecular Aging Shapes Translational Dynamics. It is about the degradation of ribosomes over time and how that changes their ability to generate proteins. You can find a link to the paper in the description of this video. We recommend reading or at least glancing at the paper before listening to our discussion of it. This is a very long paper, so a glance and a reading of the introduction is probably more appropriate than reading through the whole thing for most listeners. So, Linnea, why did you pick this paper?
I picked this paper because it's been on our list for a long time. We have heard Jordy talk of this work and think that it's a beautiful idea, but it is a long paper that we were intimidated to read. And so we had a long weekend that we were traveling, and it finally seemed like the right time where we could set aside enough bandwidth to read the whole paper through.
This went up March 9th. We probably decided to do it April 1st, and now it's May 1st when we're finally getting around to recording it.
Yeah.
And it's a delicate paper because it's a paper from one of your labs, which I don't know if we've ever done that before, one of your current labs.
Yeah, I don't think we've done a paper from a current lab before.
Is it, correct me if I'm wrong, this is Jordy's first paper that he's published before?
I believe his first author paper. I guess we could check his Google Scholar.
Right, so we have traditionally, or at least in our very early episodes, we tried to avoid doing this specifically because your first rodeo can be such a painful experience. And as, of course, for anything that's like truly cutting edge and truly interesting, there's a lot to criticize.
Yeah. But we'll send this to Jordy before we put it out, and he can veto the whole thing if he thinks it's too dangerous for him. And I thought it was worth reading this paper in depth because I admire Jordy so strongly as a scientist, and it's, good to see what him and people like him are thinking about.
So I want to start with a conversation. I already made a little dig at it in the intro of the word molecular aging.
Yeah.
And I want to be clear from like the very beginning. I am not against the word molecular aging. I think it's quite obvious that the correct word is degradation. I think that's like, you don't say that a metal is molecularly aging. You say it's rusting, right?
Yeah.
We talk about denaturing. We talk about oxidation. We talk about post-translational modification.
But degradation in biology can be a particular thing, right?
Biologists are stupid with words, and they take other people's words and make them wrong. In chemistry, this is degradation.
In chemistry, this is degradation?
Yes.
Okay.
We're talking about the degradation of certain parts of the ribosome complex. Now, here's my thing, right? This is degradation. I am fine.
I don't know if I agree that it's degradation.
Because you want to use the biology term of it's only degradation if it's been tagged for degradation.
No, I think degradation implies breaking down, like getting smaller.
So you don't think that post-translational modifications, detrimental post-translational modifications or oxidation are degradation? Right.
That would be my pushback of
You think degradation specifically, it has to get smaller?
That's what I would think from a chemistry perspective, that if a molecule is degrading, it's getting smaller.
Okay.
That you start with a long chain and it becomes a shorter chain.
So what is the word that you would use to talk about, because it's not denaturing specifically what they're talking about. Well, it could be denaturing. They actually don't know what it is in this paper.
Right. It could be that these modifications denature the protein. Right. But yeah, I mean, we've talked about this before, and this is something that we talked about.
Degradation to me does not mean breaking down.
It doesn't. Okay.
Sorry, it does mean breaking down. It doesn't mean getting smaller. It's breaking down in the way that a car is breaking down, right? A car isn't getting smaller when it's breaking down. It's degrading in its ability to do something.
Okay.
Right? So that's my pushback on your pushback.
Okay, interesting.
I think nobody would have blinked if you talked about ribosome degradation shapes translational dynamics. I think when you say ribosome molecular aging shapes translational dynamics, and then you have this science, a lot of people go, well, why are you talking about the word aging here?
Yeah.
Right? Okay, so now here, coming back to what I originally said, I think molecular aging is the right word.
Okay, why do you think that?
I think it's the right word because this is the word that gets us funding. And there's this inclination to say, hey, you shouldn't be using words just to get you funding. I think the other way, right? Like dark matter was so successful in keeping astronomy going, even though astronomy doesn't produce any product. And in this day and age where every single time you talk about your science, and people immediately are like, oh, but what is that useful for? Well, if your science has the words molecular aging in it, if I say I study ribosome degradation and how that shapes translational dynamics, I'm immediately hit with, oh, but what is that useful for? If I say I study ribosome molecular aging and how that shapes translational dynamics, people are interested. And I don't think that this is like a shallow reason to use this word. I think that this is an important and useful reason to use this word. I talk a lot about how biophysics should mean biophysics and not like structural chemistry. Because if you go in front of the House of Representatives and you describe what you're doing, your words should have the meaning that the House of Rep thinks they, like if you're a public person, you should be using the words the way people want you to. If we talk about things as molecular aging, that gets the taxpayers excited about the problems that we're doing in the same way that dark matter does. And so it actually has me thinking about how Cliff did his 2008 paper. He was talking about like biological phase separations. And, you think about it, it goes through a couple papers and the word phase separation is like quite silly, right? Like everything is phase separating. And so then he got forced into like molecular condensate and like had to get more and more specific. And actually, I remember you giving a talk at Santa Barbara and you were talking about condensates. And there's a guy in the front row who clearly came to listen to a talk about Bose-Einstein condensates. It's 20 minutes through the talk and it's like, what do you mean by condensate? Well, there's no bosons here. But I actually, like, going back to it now, I think maybe we should have kept calling them, just phase-separated organelles. Just, like, done the silly thing because it's a more of an earworm for the general populace.
Yeah.
And then we, as scientists... You could wrap your mind around the word molecular aging and like see, okay, it means post-translational modification, which means anything, right?
Yeah.
Or it means oxidation, or it means denaturing, or there's many things that it could mean. So in the end, I went through this whole paper highlighting in red every time it says molecular aging and writing degradation. And at the end, I was like, actually what I want is not to call molecular aging degradation. Let's call it molecular aging. I want to find other things that I can call molecular aging, and I want to talk about structural aging, and I just want to like tack the word aging onto anything that has any time component to it at all that doesn't already sound attractive to the general populace.
Interesting.
Because I'm really interested in aging, right? I went and took this whole
in
course.
Organismal aging.
I'm interested in organismal aging, right?
Yeah.
I went and took this course in Woods Hole to learn more about where the field is. I get very excited about aging. I saw this paper and I was like, that's not really aging. And I went further through the paper. I'm like, okay, it's borderline. Maybe it is really aging. But the point is it got me excited. It'll get other people excited. And I want my work to get people excited about aging. I am very shy about the word aging. Molecular aging is a great hack. It's something that you can like say the word aging without being shy. I'm studying something that's equivalent to studying nuclear weapons, right? Like, I don't think people are afraid enough about like aging study and AI studies, and like people are afraid of nuclear studies. Good. Apply that fear elsewhere, people.
Interesting. Yeah. So, I mean,
So we gotta, instead of for active matter, we gotta start studying structural aging of active matter.
Structural aging. I also have been thinking about whether molecular aging is the right way to talk about the changes that are happening to these structures. And the parallel that I draw is to carboxysomes in cyanobacteria. We've done some journal clubs on those in the past. And they stick around for a long time as well, like multiple generations of these cells. And can we call that molecular aging? So like a carboxysome could be molecularly aged. And there the connection to real aging, to organismal aging, is not present, right?
And I think that's fine. I think we should call that molecular aging. I know in that other episode I said we should not call that molecular aging. And we talked about this paper in that episode because this is the borderline case where it's like, we really are getting very close to organismal aging in this paper. I think we should call that molecular aging. I think we should use that word freely. It's a fun word that captures the public imagination and helps you with the immediate question, what is that good for? What is the engineering? What is the product? How does this interface with capitalism?
Yeah, I'm pretty convinced by your little spiel here. We'll see what the listeners think.
So one thing that I'd like to talk about, just going from the beginning, is they use halo tags to label either new ribosomes or old ribosomes. So they either block everything and put in some halo tag. So I guess I have two questions here. One, what is the thing that's actually fluorescent in these experiments?
Yeah, you do some like click chemistry to bind a peptide to the halo tag that has a fluorescent tag on it.
So these cells are being incubated with a dye. Is that correct?
That's correct, yeah.
Okay.
Like a Genelia floor.
Yeah, so that leads me to a very small concern for like something like figure 1G, where if it's getting brighter and it's being incubated with a dye, it's hard to know exactly what that means.
I think you're not incubating the whole time.
Yeah, just my experience with dyes is like you wash out the dye, but you still have like dyes tend to get brighter over time, and canceling out for that is really, really hard. That I had like an incredible difficulty convincing myself that my ATP red 1 signal was actually going up, and it wasn't just more dye getting into the right places. So
Yeah, this is not a tissue.
Yeah,
So
that's true.
I think this is a lot easier to work with because like there's no like intercell space that the dye could be hanging out in, right? If you wash out the whole media.
Yeah, that's very true. Yeah, it is. My second question, which is really the question I wanted to go for, is did it have to be halo tag? Is this an experiment that is only possible due to the halo tag technology? Or has this been possible for longer than halo tag? So you're shaking your head no, that this is a halo tag specific technology.
I think so, yeah.
What is it specifically about? Like, so then you're saying that this couldn't have been done via like streptavidin and biotin.
Well, that's not a live cell.
You can express biotin.
Right, but you can't do the streptavidin biotin chemistry in a live cell.
Okay. And so this like lock and key mechanism of halo tag just really didn't exist before halo tag.
Right, like halo tag, snap tag. So you couldn't do, I mean, you could do like, are there fluorescent proteins that are photoconvertible?
Sure. Yeah. Like the Dendra series of fluorophores are photoconvertible.
So I think you could get close to something like that with a photoconvertible fluorescent protein, but I think this is a neater way to do it. It gives you a lot more flexibility, and then your photostability is way stronger. And
yeah,
I think the tag is actually smaller.
Question though, like why? Okay. So I hear you that it wasn't, but you could have done this with a photoconvertible dye. Yeah, why do that instead of this halo tag block pulse chase? So what they do, for somebody listening, is they've got the ribosomes expressing a very fancy tag called a halo tag. And that halo tag, essentially, you can put in a dye that will snap onto that halo tag, or you could put in a blocker that will particularly block this halo tag. So you can imagine you start your experiment, you put in the blocker, and then you wash out the blocker, and then you put in the dye. So now the dye is going to accumulate only onto new ribosomes because all the other ribosomes got blocked. And so that's a way to trace the ribosomes, and specifically to trace the new ones. You could do the inverse and just trace the ribosomes that already existed and block the new ribosomes. And they use this mostly as a way to purify ribosomes of particular times.
Yeah.
Is mostly the way that they're using this.
So for instance, I think you just answered your own question. Like that experiment would not be possible with a photoconvertible dye.
I guess the point is the purification is not possible. That's the big thing. Because actually like seeing what is old and what is new is completely, I mean, it's nice, but it's useless, right?
Yeah.
It's all about purification.
Yeah.
And it's really the purification that the halo tag enables. And you're saying there was no technology. I mean, you said snap tag for a second. Isn't snap tag older than halo tag?
I know that halo tag is like 5 times more efficient than snap tag. I don't know which one came first.
Okay.
But
yeah.
And they're not new, right?
Right.
Like, I mean, I'm sure the paper is cited in this. I would say they're 10 years old, right? Like Pooja was using snap tag in our PhD.
That's why I say that halo tag, I think, is newer.
I think they're
halo tag 2008.
Yeah, they're pretty old.
New by it's almost 20 years old now.
The fluorophores have gotten a lot better.
Yeah, the Genelia floors and all this.
So like the labeling and the reduction of the background and the ability to get these dyes into live cells has really improved. And so I think that's probably why this is like now possible, whereas it wasn't 10 years ago.
You often talk about that you really want beautiful science, that you want to see beautiful images. This paper has some images. I don't 100% know. Like, it's nice that it has images. I'm not complaining about them. It's definitely not the crux of the paper.
Yeah.
Really, this is a graph paper.
Yeah.
Do you find the images in this paper? Are you like, oh, that's nice. That's a little bit of beauty.
I don't find a lot of beauty in I think it's a beautiful paper, but I don't think that the figures are
I'm talking
beautiful.
about aesthetically.
Aesthetically, I don't find watching the ribosomes beautiful. I think the closest you get to that is in the nucleolus stuff, like Sophie's old papers. But even then, I feel like that's a big weakness of the ribosome biogenesis field.
At one point, they talk about these traffic jams where a ribosome that has molecularly aged.
Yep.
They have a better word for it at some point. Where they say that it's been in like residence too long. And I love that like concept of like the ribosome is like a musician in Las Vegas that's having a residency or something, right?
Yeah.
A ribosome that is molecularly aged is moving along some RNA slower than the ribosome behind it. The ribosome behind it is like, come on, move out of the way. It's like an old person driving a car, right? I wonder if there is a video of that.
Of that happening.
I can do a gliding assay
Yeah.
of kinesin on microtubules. I put down the microtubule, I watch the dots that are the kinesin move along the microtubule.
Yeah, I feel you could do something like that with ribosomes.
I want
right?
RNA gliding assays with ribosomes.
Okay.
I actually really want that you watch the ribosomes migrate along the
Yeah,
RNA.
do people do this? I don't know enough about the ribosome field. One of the things I learned in the course of this project was that the ribosome large and small subunit don't travel together.
Yeah, that they come apart.
They
and
come
back
apart
together.
and then
Yeah.
they come back together. So you can have, and they talk about this in the paper, that these large and small subunits have different aging pathways.
Talk about naming things where like rather than large and small subunit, you just call them 60S and 40S. Boo.
Oh, you're upset about the names.
I'm not upset about the name. I'm just like 60S and 40S is like monitor naming territory, right? Where you're just like a jumble of letters to describe the monitor that you just bought from Dell, right?
Yeah, I think it has something to do with the length of something in the...
One of my theory group is actually on this paper.
Yeah.
Andrej, Košmrlj, and Qiwei are on this paper.
Yeah, what did they do?
Yeah, so you're like, where is there any physics theory in this paper? Where is it? Okay, so bottom of page 4. This is my guess at what they did.
Okay.
All right. It says to understand the origin of ribosome dynamics, we consider that the time rate of change of the ribosome concentration R should be governed by the equation that the time derivative of R is equal to the ribosome production rate minus the ribosome degradation rate times the ribosome current concentration minus the dilution rate times the ribosome concentration, where the dilution rate is due to cell growth. It's a super simple equation.
Yeah.
I've changed the wording a little bit there so that it reads better for people who are listening to this, that you would learn in like a first semester physics class. That's got to be it.
Are you sure? That seems like such a Cliff equation to me. Like,
It does say, if you go down to the bottom, because I was like, what did Qiwei actually do here?
Yeah.
It says that the theory was done by Qiwei, Andrej, and Cliff.
I feel like Cliff wrote down this equation and then maybe they had to do something
They
with
did
it?
do a tiny bit of cleverness here, which is that they say, look, the solution to such an equation, to such a differential equation, is an exponential
Yeah.
with a time over a characteristic time. They say the characteristic time for this solution is going to be the degradation rate plus the dilution rate. That's the answer to the equation. Sounds good? The clever thing they say is we can experimentally measure the dilution rate because we know how much bigger the cell is getting. And we know the answer to the whole equation because we are able to watch ribosome concentration over time as well. Therefore, we can get out of this the ribosome degradation rate. We can get K degradation from this equation, even though we can't directly measure it. And we find out that it's actually roughly the same as the dilution rate. That seems clever.
Yeah, I didn't look at figure S3A. One of the things that bothered me in this paper was that they don't actually report the numbers.
Yeah.
So like,
But
of their fits.
What's the point? Why do we need the degradation rate? Why does this matter? For what this paper is actually about, why are they doing this at all?
Why are they doing this theory at all?
Yeah.
No, they do give this. Okay, time scale here. Sorry.
Yeah, they give the time scale.
They give the time scale.
They just don't give K degradation.
Why do we care about the degradation rate?
Yeah. Or
Well,
why do they care about this theory?
Well, I think you care about the... Why do you care about the theory? Okay, so what does the degradation rate get you? So the tau they're saying is some which they experimentally measure,
How they measure.
is some combination of the dilution, which is something they're not interested in, or like is just like background, and the degradation rate, which I think is what they are interested in.
Plus the production rate. But they're not interested in the degradation rate.
Well, isn't that the whole, like, if the timescale of the degradation rate will give you some insights into what is molecularly aging these things, right?
But they don't really, like, state what they think is molecularly going on with the ribosomes. Like, they state what the ribosomes start doing worse. The point of the paper is, and we'll get to this in a second, but the point of the paper is that the ribosomes start doing basic amino acid sequencing worse, right?
Yeah.
That's what they get into. They don't get into, it's oxidation, it's denaturing, they don't get into, like, how it is itself degrading, aging.
Yeah, sure. I mean, I think this gets to a kind of fundamental thing that's still being worked out in this paper is like, what is the core finding of this paper? There's a lot of data, right? And I'm going to keep going for a second.
Okay, sorry.
To me, right, like they're arguing that these ribosomes age over time, and that's the thing they're trying to unpack in this paper. And so giving a time scale for that aging is important, and saying what the contributing factors are.
But this does not give a timescale of the aging. This gives a timescale of the decrease of concentration of pre-existing ribosomes. This gives you a timescale of ribosome recycling. And they do not report on K degradation specifically. They don't like say that number ever.
Yeah, okay.
And so again, I don't see any point to this theory here. This seems like a non sequitur.
Yeah, they just say to understand the origin of these dynamics. So to understand the origin of the time scales.
Yeah. And I wouldn't have a problem with it, especially if it was like off into the supplement, except for it makes its way into a lot of the graphs, into a lot of the figures. It makes up a lot of the graphs in the figures.
I just feel like this is related. One of the questions I have written down in my notes to talk about. So we like read this paper a while ago.
This is the biggest gap between reading the paper and discussing it that we've ever had by a long shot.
Yeah. So I'm not totally in the same headspace, but one of the questions that I wrote down when I was reading this paper is, what is the average lifetime of a ribosome? And what is the spread of lifetimes? And I think that these
numbers are
giving
average
you that
lifetime of a ribosome, but due to ribosome recycling, that the ribosome is getting eaten up, right?
Yeah, so that's how long a ribosome is sitting around and can potentially get aged or become modified.
Yeah.
Right? And I feel like that's an important time scale to be thinking about when considering, like how long are these things sticking around and how long do they need to be sticking around? And then also in the end of this paper, right, they start talking about organismal aging. And so like how long are those ribosomes
if you wanted
sticking
to
around?
do that, you would have just fit it to an exponential decay and reported on the characteristic time.
Yeah, they did that.
Yeah, but here they go one step further. They create this equation.
And I think they did that because they could. This is like such a Cliff thing of he's just like, we have this data.
So that is my objection. Is that if it was just this block of text, I'd ignore it. It makes its way into the figures. The figures are already really, really hard to unpack. I had a lot of difficulty with this paper's figures. I needed that information to go off into the supplemental.
So you're upset that they say that these are fit to exponentials in the figures? I feel like they didn't include this enough in the figures. My complaint was that like it took me a long time to find the tau. Like I wanted them, when I look at 1K, I want tau equals 30 hours. Like I don't just want to know that it's fit to an exponential. That I also like, even without reading the text, I can see that this is an exponential decay.
Yeah, I just felt like this was unrelated to the main thesis of the paper.
Which is that the molecular aging of ribosomes causes these preferential defects in...
Yeah, the inability to deal with certain amino acids in a timely manner is, I think, the main thesis of the paper. I think it's a strong thesis. I spent a really long time trying to figure out what this paper was about, and finally deciding that, look, this is what it's about, is that ribosome molecular aging compromises elongation dynamics at basic regions and increases collision propensity. And by basic regions, we mean basic amino acids. It screws up on trying to translate basic amino acids. It gets stuck. Other things slam into it, and it screws up all of its translation dynamics.
Where basic is a chemical property, not a
This is the
simple
finding. Like the finding of the paper is twofold. It's that the problem is intrinsic to the ribosomes. It's not the system making ribosomes' life harder.
Yeah.
and that the problem is the ribosome's ability to deal with basic amino acids. That's the paper. It only really hit me when they were in the discussion. Recent studies have reported translational defects that intensify with organismal aging in systems such as C. elegans. In these models, organismal aging exacerbates ribosome pausing at regions rich in basic amino acids, promoting ribosome collisions and driving the accumulation of incomplete nascent polypeptides. And I read that in discussion. I was like, wait, if that was already known, what is this paper about? And so I think what this paper is about, if that was already known, is the problem of this basic amino acid translation is intrinsic to the ribosome itself.
Yeah, and not to the other properties.
And that's the main finding. The ribosomes, or there being something going on with ribosome aging was already known.
Well, I think what was already known was that aged organisms have something going on with their ribosomes.
Yeah, that's what I tried to say.
Okay.
Yeah.
Not that ribosome molecular aging.
Right.
Yeah.
And so I think the key takeaway from here is that it is an intrinsic thing to the ribosome itself. Here we can show this little thing that's going wrong. And then it turns into like a classical biophysics paper of
it's such a biophysics
there's
paper.
some like structural chemistry that makes me want to kill myself.
They have these like, I hate them, figure 3 CHI with the amino acid letter size. And it's this kind of data that we were not raised on, right?
I think I was more raised on it than you were. That I was encountering this data consistently throughout the beginning of my PhD because I was housed in a molecular biology building and I was routinely going to molecular biology talks.
Sure, but the data that we present in our papers is often related to microscopy.
I would say that's more true for you than it is for me.
Okay.
That you often say that you like really love microscopy data and that if you can't see it, you don't like love it.
Well, but I run gels.
You run gels. I hate gels. I hate gels. I hate gel machines. I hate the word gel. I hate pictures of gels. I hate the biophysics of gels.
There are gels in this paper.
I hate them. You like the unit of bits on those graphs?
No.
This paper
like
has a
units.
lot of y-axes that don't have units, as does any biophysics or any biology paper.
Yeah.
But it has made me start to, not this paper in general, but I think just like during this paper, I think a new pet peeve of mine is graphs where there's a log of something and that log is not something divided by another thing. That log is just being used as a weapon when you don't have units. And it just like makes it impossible to figure out what the units of anything could possibly be because the log demands that you don't have units.
Yeah, I wasn't upset about those.
They do the following experiment. Together, these results demonstrate that shifting the ribosomal pool towards older ribosomes is sufficient to amplify molecular age-dependent translation defects and suggest that organismal aging and disease-aging perturbations in the biogenesis and turnover of ribosomes could similarly enrich aged ribosomes. This is the bottom of page 14, you're looking at me. They shift the ribosome pool towards older ribosomes and see more jamming.
Yep.
As far as I can tell, and again, it's a long paper, maybe I skipped over, they don't shift the ribosome pool towards younger ribosomes. The way they do this, right, is they halo tag the older ribosomes, and then they specifically degrade anything that's halo tagged with this PROTAC treatment.
Yeah.
So they can do it the reverse way around, right? Or sorry, they specifically degrade anything that's younger, so they have only old ribosomes. But they could end up with only young ribosomes as well, and see if translation defects shoots way down below the norm. And they don't do the reverse experiment, right?
They don't do the reverse experiment. I agree. I didn't consider the feasibility of the reverse.
That you halo tag.
Well, so it was this PROTAC thing, right?
Yeah, so you halo tag the older ribosomes, then you put in a blocker so any new ribosome doesn't get halo tagged. Then you use PROTAC to degrade all the older ribosomes instead. And now you just have super young ribosomes, and you find out how does the cell behave when we have just super young ribosomes. Because they can always do the pulse chase.
Wait, but I don't understand how that works. The PROTAC is degrading the stuff as it goes. So how are you degrading only old ribosomes? Like, won't you just degrade all of the ribosomes in the cell?
PROTAC degrades anything that is halo tagged, right?
Yeah.
So you can choose to either...
But that will kill the cell if you treat it initially.
So you put the PROTAC in after you've either tagged the young population or the old population.
No. Halo tag is always there.
Yeah, but PROTAC doesn't have to always be there. Isn't it just like a drug that you're putting in?
Yeah, but the halo tag, the ribosomes are always have a halo tag on them. pre-existing 6DS ribosomes were first saturated with the halo-tag TMR ligand, preventing subsequent labeling. Okay, so you're saying instead of that, you would start by treating with Protac?
I mean, how does Protac work? Because I'm not quite sure I understand what your confusion is. Protac attacks something that has a halo tag ligand open, right? And you can always block the halo tag, right? Like one thing we have proven in figure 1 that we can either
So you block...
fluorescently tag
Yeah.
old ribosomes or fluorescently tag
Sure.
young ribosomes.
Yeah, you can do that.
Okay, so if I can fluorescently tag young ribosomes or old ribosomes by choosing when I am fluorescently tagging versus when I am blocking,
Yeah.
why can I not, instead of attaching a fluorescent thing, attach a eating you up thing?
Well, because I think the thing is eating you up immediately.
Immediately when I add it, I decide which things I'm going to block first, right? I can either block the old ones or the young ones.
No, because to block the young ones, you have to have labeled the old ones and then not labeled the young ones, right?
So to block the young ones, oh, I'm starting to see what you're saying. Yeah.
So
Okay.
if you degrade all the old ones,
Okay,
you're
so
just
wait.
killing yourself.
So to label the young ones, we put in block,
Yeah.
we block all the old ones.
Yeah.
Then we wash away, then we put in a label and we get labeling of only the young ones.
Yeah.
To label the old ones, we
Label,
label,
wash
then
away,
wash away and we put in block.
or nothing, right?
Yeah. Okay. So to kill all the old ones, we'd put in the Protac, it'd eat up all the ribosomes, the cell has no ribosomes anymore.
Yeah. And then
And it
it
just
dies.
Okay, yeah.
So I
Seems
think
very
the
good.
reverse experiment would be really hard.
All right, it took us a long time to get there, but I got there. We need some light-activated Protac. Where did all my comments go? That was useful. I understood something there.
Yeah.
So then the final thing that they do is they do it in C. elegans.
Yeah.
And here I think the language gets a little bit... They talk about that the ribosomes, when they're not working perfectly, they call it translational defects. Okay, I think that word becomes a little bit confusing, but used in isolation, it makes sense. The problem is then they go to a whole organism and they manage to isolate the old ribosomes and the young ribosomes in the organism. They do this on a totally different timescale than in the cancer cell. They do this on an 8-day timescale.
Yeah.
But I'll let it go because it is cancer cells versus an actual organism. There's no reason to think that any of the timescales or any of anything is going to be similar.
Yeah.
So, whatever works, that's fine. But then they say that there's reduced translational activity in the organism. They're doing this based off of what the ribosomes look like, not based off of what the proteins. They don't have a proteome of the C. elegans. And I think at this point, I really do want a proteome.
Oh, yeah?
Like I understand.
Don't think that gets at their question, right? Like, I'm
No,
sure there's a
gets at
proteome of a C. elegans at day 2 and at day 10 out there.
Right. But
And that the absolute protein levels don't change.
But if they don't change,
Yeah.
and you're saying the ribosome has translational defects, which I don't love that term, right? If they don't change, then what are you really saying? We talk about function in biology, right? That it has to have some critical function. If this change hasn't had any function, what are we talking about? And I do think, I think you'll see a difference. I think if you get a proteome of a C. elegans, you'll see, hey, these proteins with these more basic amino acids are not as expressed as they used to be.
Oh, yeah? You don't think that those proteins are just older? Getting to other molecular aging questions?
I mean, but I think if you want to make this kind of claim, there starts to be some dancing about words, about the age shift of the ribosomes contributing directly to altered translational dynamics during organismal aging, which I think is it's too big of a claim for the data that they have. It is the last figure of the paper, so maybe that's acceptable. That is the time to make too big of a claim.
Yeah, I mean, I do think that this is a stretch figure, and I know that it's something that they...
It is the last figure. I do think it is well known and appropriate for the last figure to be a stretch figure. So maybe I'll dial back my criticism.
Well, I don't know. I mean, I guess I don't totally understand what your complaint is. So I see in figure 6B.
I think my complaint probably comes down to the word defect, which is that when I think about an organism having a defect, right, that really to me is a functional
But
defect.
I don't see, in the figure caption, I don't see the word defect. They
Sure.
say ribosome molecular aging shapes translation dynamics during organismal aging.
That's
is
true. They
it
don't use it in the titles. They use it throughout the paragraphs.
in the text, they say defect.
Yeah, so like page 16, ribosomal molecular aging shapes translational dynamics during organismal aging. Given the translational defects associated with molecularly aged ribosomes, and they've been using the word translational defect throughout the paper.
Yeah.
Up until the point that they started talking about an organism, I was on board with it.
But here they're still talking about the molecular side. They've
Yeah.
put it in the organism section, but this phrase is still about the molecules.
Yeah, yeah. So I made a comment here where I said, I'm not super comfortable with this wording of altered translation and defects. Using the word dynamics makes it fine. It's not wrong, but it's like 2.5% wrong. I think there's this little niggle that this can be very easily misinterpreted,
Well,
but it
so
is a stretch figure.
This niggle, right, I think is the problem with molecular aging. Of like,
Yeah. It's true that I made this whole pitch for why we should all say molecular aging, but it does cause you to start planting flags where you shouldn't plant
starts
flags.
to muddle a little bit what's molecular aging and what's organismal aging.
Yeah.
You have to be super precise with your language, and I think they are, but it does still have that niggle feeling, right?
So normally we ask this question, if you could have one thing added to this paper, what would it be? For me, I would have proteomics of a young, is it just a mammalian
I
cell?
think it's just a mammalian
All right,
cell, yeah.
of a young U2OS and an old U2OS. And I want proteomics of a young C. elegans and an old C. elegans. And I want that just shown either way. Does it refute the story? Does it support the story? Or does it just look like nothing and that just is what it is?
Yeah, I mean, I think anything...
I want that in the supplement. Maybe it already is, because who knows what's in the supplement?
Who knows?
Because we're now on page 17 of this 55-page paper. I definitely didn't look at the supplement.
I did not either, and we had like a month with this paper.
If Jordy is listening to this, this comes from someone who has written just monster paper after monster paper. You and me got to stop writing monster papers.
I think, yeah.
These are unreadable.
So that's what you want to add to this paper.
Yeah. I do wish, again, this discussion makes me realize that like a lot of things that they're talking about here are already known and already thought about. And I think some of my confusion or some of my niggles come from not understanding the surrounding literature.
Right.
Jordy getting into this has made me really fascinated with the ribosome. We've talked about the ribosome a lot. We tried to listen to his talk at ASCB, but we had Asya, so I didn't get any of that talk. I just sat in the back trying to keep Asya quiet. But it seems like a really fascinating thing to study.
Yeah,
if you
so
could
I
add
am...
anything, what would you add?
What would I add to this paper?
Ribosomes that have undergone extended residence in vivo. This is my favorite line of the whole paper.
Yeah. I wanted to say that this ProTac thing seems pretty cool. Would you use this ProTac thing in your system?
I would first have to think about a way that I wanted to use halo tag. I remember when we first came to Princeton, we were talking about some halo tag experiments, and then my eyes rolled up inside of my head and I passed out.
Well, so these pulse chase experiments like are the bread and butter of Cliff's lab these days, and I still think we should do them in the Jonicus lab with Clammy. Like you could pulse chase label Rubisco and understand a lot more about how it gets into and out of the purinoid.
What am I supposed to do for this for a multicellular organism?
Well, so I don't know how if ProTac's been used in a...
How did they do this in the C. elegans?
This is
They
all...
did do a pulse chase in the C. elegans.
Like you can put halo tag in the same way But
I never think about the time dependency of any organelles. Like I do really want to know if mitochondria migrate from cell to cell in a tissue. And then I really want to know, because they do, it's just not reported. I really want to know to what extent they're migrating from cell to cell, right? And if there's signals that are like letting mitochondria move in giant paths.
Yeah, I mean, there's a lot of questions, and maybe this is all
But
known
this is
about
really
mitochondria.
a job for photoconvertible mitochondria, which is what I've been doing. The halotag thing really shines when you start to purify the protein. And I think I've said this before to some fame in your lab. I would rather pull off my fingernails than run any more purifications in my career. That I'm just...
These ribosome purifications are
They're rough.
Well, yeah, you have to do a sucrose gradient and a very long spin. So it's a tough.
Well, how many
one.
days is it?
Oh, I think it's about as long as a protein purification. Depends on how hard you
here's
go.
my question. Should I be thinking tubulin prep or should I be thinking kinesin purification?
I haven't done a ribosome one, so I can't say for sure if...
At one point, I started to hear that people had ways to purify mitochondria, but I think you always end up disrupting the outer membrane when you purify mitochondria.
But
I'm not so excited about it.
So you're saying that you would just use a photoconvertible dye?
To watch spatial migration, yes. The main thing that we talked about at the head of this episode is that the beauty of the halo tag really is that they can then put the halo tagged ribosomes on a column, sort them, purify them out, and then really go and look at what is the structure and what is going on inside the ribosome. And that's what the paper is. That's the meat, that's the science, that's the PhD.
Of this paper, yeah.
Outside of that, it does seem totally acceptable to just use a photoconvertible ribosome or like Dendra 2 attached to a ribosome.
Yeah, I just think that those are much harder to... their brightness is much lower, but if you're not limited by brightness, then sure. But the other power of the halo tag is this PROTAC thing. That's what I'm trying to say. Is that you could selectively degrade a protein.
Yeah, again, I don't see where that's super helpful.
I mean, so again, going back to the carboxysome system, which neither of us work on, but you could selectively degrade old carboxysomes.
No, maybe I do see how it's helpful, right? I can right now go after my mitochondria with this drug or that drug.
Yeah.
Or go after my glucose uptake transporter with this drug or this glycolysis enzyme with this drug. But each time it's each drug has something finicky about it. Each thing is a little bit of a weird perturbation. It would be nice to go after everything with one tool, which is I genetically modify my embryo, which I'm pretty good at these days. I genetically modify my embryo to have this halo tag thing, and then I PROTAC it. I guess I'm worried about diffusion coefficients. But again, they do it in planaria. But planaria are pretty easy to get things into.
Anyways, that was a cool...
Yeah.
Degrading new protein. I think that's a new
cool
tool or a tool that I wasn't aware of yet.
Yeah.
So I think that's pretty cool.
Bye. Bonus content. Linnea, as soon as we ended, said she had some negative things to say. So we're just going to record the negative things just in case. And maybe we'll put it... that we've often talked with people about putting on Patreon, the unedited versions of things where it's just negative the whole time.
I was confused about this section that starts talking about this pseudouranine 18S to 210.
Yep.
Why are we in this? And
well, one could argue that this is the real...
That this is the real finding. If this is the real finding...
Well, it's like this hourglass, right? You start big, you zoom in, you zoom in, you zoom in, you zoom in so much that everyone's screaming besides you, and then you start zooming out, right? And I think they do that really well.
Yeah, I mean, if this is a true biophysics, in the structural biology sense of the word, then I see that.
My justification for Psi 18S-210, which is the thing that they dive really far into, is that this is a biology paper. And so they do need to provide mechanism. And without this Psi 18S-210, there is no mechanism provided in this paper. Mechanism is a bullshit word that biologists use because they don't think, right? But generally the way I like to think about it is you have to report on a thing, and then you have to report on a second thing which explains the first thing. And they call the second thing which explains the first thing mechanism. So you always have to report on two things in any biology paper or the biologist won't listen. And so Psi 18S-210 is the second thing that they report on that explains the first thing, which is that ribosomes do not do basic amino acids that well after they've been aging for a while.
Is that really shown? Is it... I mean, maybe I didn't follow closely enough.
The analysis revealed a selective depletion of 40S ribosomal subunits, harboring the conserved pseudouridine Psi 18S-210 within ribosome collisions driven by molecularly aged 60S subunits.
Okay, so they're saying there's less of these with the aged 60S subunits. So
they're suggesting a potential role in modulating local structural integrity or protein RNA interactions. And they have to do this, not necessarily because it's good science, although I'm not saying that it's bad science either. But they have to do this.
It just doesn't seem to me that it explains
because they have no explanation, right? They say in their discussion,
Yeah.
defining the precise molecular features that distinguish ribosome age demographics and drive age-dependent functional dynamics will require further investigation, but likely include time-dependent protein composition and post-translational modifications or stochastic chemical damage such as oxidation or hydrolysis. So they say, we have no idea why this ribosome is acting worse over time.
Yeah.
In a biology paper, people will say, reject, they have no mechanism.
But
so the Psi-18S-210 is their shield. And if they don't have it, I don't see another shield. Do you see another shield that they can use here?
my complaint with this shield, I hear you that they need a shield.
Yeah.
My complaint with this shield is that it is a small shield. Like,
It's a small but completely indecipherable. Like, it's got a really weird name. I skipped over most of this section. I know you skipped over most of this
well,
section.
I mean, I have it highlighted in red. The whole thing is red.
But I would say if you are doing a mechanism shield simply because you're like, okay, biologists are idiots and they cannot have any single finding at a time, they must have two findings at any time, then the more indecipherable your shield, the better.
Together, these findings identify Psi-18S to 210, or Psi-18S-210 negative ribosomes as molecularly vulnerable ribosome subpopulation that become collision prone in concert with molecular aging of 60S ribosomes.
Yeah.
So this isn't even about the aged ribosome.
Yeah.
It's about the partner of
Yeah.
the aged
Yeah,
ribosome.
I'm not saying that it's good. I'm not saying that I like this part. I am saying that this is a mechanism.
Okay, and you think that somebody said you have to have a mechanism. in
I'm
this
saying that
paper.
without this part, it is not clear to me that they address what is the mechanism. If they get hit in review with no mechanism is given, they will be rejected immediately. There's no defense against that in review other than to give a mechanism or to say, hey, they missed our mechanism.
You don't think that this section makes the paper weaker?
Oh, I do. But I think that this is the problem with biology, is that every single biology paper has a section that makes the whole paper weaker because they must have two findings in any paper, a actual finding and then a second shitty finding. You don't have that in your paper?
A second shitty finding?
What is the mechanism in your paper?
The mechanism is Qiwei phosphorylates EPIC1, and this disrupts phase separation.
Right, that's the finding. What is the mechanism by which it phosphorylates to EPIC1?
It's a kinase.
My paper
I
is
don't know what
littered
you
with
mean
these.
that
My
the-
paper is littered with these bullshit mechanisms to try to defend
I guess
against.
we have this section about the Ribisco binding motif. Maybe this is your mechanism.
Yeah.
Where, like, how does the kinase phosphorylate EPIC1?
Yeah, that's exactly what I'm asking. See?
Yeah, I also hate this section in
Yeah,
my
see,
paper.
they're guaranteed to show up in any biology paper because these people are, I don't know what goes wrong with everybody's brains because it's not like the physicists are any better these days.
and maybe it's just something we don't understand on a cultural level, but it's, to me, this section, I was like, I don't think this needs to be in the paper.
Hey, you know what else doesn't need to be in the paper? This equation.
Maybe what happened is Cliff was like, I bet we could write this equation. And then Jordy asked Qiwei to write it.
Yeah, are they friends? I don't know that
don't
entire
know.
generation.
Yeah.
Like, we keep inviting people over only to find out that like A hates B in that generation or B broke up with C, and we're like, yeah.
We're old now. Yeah.
I don't know who's dating
When I thought
who.
a 24-year-old and a 30-year-old were in the same generation, that's when I was like, oh, I've just gotten old. Anyway,
I should ask Andrej because that's the only like
there's nothing in the
physics.
supplement.
That's the only equation I see in the paper. But again, I didn't look at the supplement. Jesus Christ, how could I possibly make it to the supplement? It's a 55-page thing. Oh, there is some. There's the same equation. There's the same equation.
Ribosome turnover
Yeah, yeah.
analysis.
Yeah, I mean, that's really it, Lenia. That's really it in this paper.
But they didn't, maybe they helped with the...
Experiments, yeah. That's my thought, too.
Or the data processing of the sequencing and
You
stuff.
think that they helped with Go terms?
No, I don't. I mean, I'm sure Troy did. I'm sure that's why he's on the paper.
There's no way that they did any data processing
Okay.
help. There's got to be those two equations.
Interesting. This isn't a bad thing, but it's a question that I had.
Okay.
In the introduction, they say, assembled within the nucleolus through a complex high-flux biogenesis pathway that produces thousands of ribosomes every minute
Yeah, yeah.
in proliferating cells, This process can consume up to 60% of cellular ATP in eukaryotes.
Yeah, I knew this.
You knew this?
I knew this, yeah.
And these 27, 28, they're real papers. How do they measure it?
This is a well-known thing.
Okay.
I've never looked into the original papers that made this measurement, so it could be that it's bullshit, but I've never thought to call bullshit on it before. We could look into it and like find out how they measured it. But yeah, essentially, right, you can just presumably you can just look at a cell that's doing this and a cell that's had this inhibited or is not doing this and look at the difference
in ATP.
In budget.
Yeah.
But yeah, it's like, the two that I always have in mind is that a cell that's going heavy on ribosome production
is a cancer cell.
No, can use up more than half of the ATP of the cell. And a cell that's going heavily on actomyosin migration can use up more than half of the energy budget of the cell. Those are the two like big things in my brain that can dominate the ATP budget when it's happening.
Like if you produce more ribosomes, the ribosomes are doing more protein creation.
Yeah.
So is it the ribosome biogenesis that takes up so much energy? Or is it that just having a shit ton of ribosomes takes up a lot of energy?
Yeah, and also can you like shut a ribosome down, or once you have it, is it stuck just doing things forever?
Yeah.
Does a ribosome use up ATP?
I don't think so.
What is it? How does it do its job then? It has to use up ATP.
Yeah. This is like a pretty big blind spot.
A ribosome must use up ATP, otherwise, or it has to use up something. It could use up GTP. It's got to use up some energy source.
When would it use it? It uses it so it binds. That's a thermo process, right? And then it pulls the
they use GTP.
GTP. Okay. And they do that every time they like bind to an amino acid or something, or to attach
Ribosomes
amino
require
acids
GTP
together.
to catalyze the peptide bond formation during translation.
Okay. So to bind the two amino acids together.
And then ATP is hydrolyzed by enzymes such as ABCE1 to split ribosomes after translation is complete.
Ah, interesting. Okay.
So there is some ATP use, but it is, I always like think, sometimes when I like don't have ATP clearly in my mind, I'm like, I bet you this is one of those GTP processes. Because then, like GTP falls out naturally, right? Like you get GTP naturally. About 10% of the ATP you make is actually going to be GTP. So about 10% of the processes in the cell are just going to use GTP because something's gotta.
Okay, those were my final comment.
Yeah, bioenergetics of ribosomes, TOR1, learn about growth. This is what people really thought that I was doing in 2022, 2021. When I would like give these talks, they were like, oh, you must be looking at lipid production, ribosome production, TOR1. I was like, oh, actually, I'm doing none of those things.
Yeah.
But that was
ribosomes are interesting. But I also, there's a whole, they get too close to genetics for me.
I guess one thing we didn't talk about that I really meant to talk about is this seems like all about drug targets, that ribosomes are a really good drug target. Yeah, it seems like a really cool, very jealous of Jordy and this trajectory that, we were at the end of our PhDs. We're like, what have we been doing for our whole PhDs? This is like, I've been doing this thing that I could patent and get a lot of
I think that Jordy is so over this.
He's gone the other way. Now he wants to go do something purely aesthetic and like fundamental biology, physics. Yeah. Although I wonder if they care about physics, right? This is really bioengineering. This is like right up against the wall of interfacing with capitalism. That like I really was reading this out, and like this is an actual question I have to you if we're not recording. Are they patenting some of like the ways that they have?
I think they say in the acknowledgments that they are like conflict of interest.
A patent application covering the work described here and is currently pending. Yeah. Because this really seems like directly headed towards seeing if we can do like anti-aging treatments. Which is scary. Again, it's like nuclear research. I wonder if we should cut some of this bit and put it in. I don't know. Probably not.
I say we send it to Jordy.
Yeah.
And then he can say, cut more, please.
Yeah. All right. Now, real bye.
Okay.
Bye.